Good afternoon, Year 12. So from the feedback I've had, it looks like you're enjoying these lessons more than simply taking notes out of a textbook, which was probably predictable. So I've decided that for the duration of the time that you're out of school, and hopefully that will only be this one lesson, that I'm going to continue making these. We're going to continue with ATP today, which is the next stage in the biological molecules topic. You may have had some teaching on ATP while I was away. However, from what I understand, some people felt that they didn't understand ATP at the end of the lesson, they were left behind. And what I don't want is for anybody to not understand this part of the topic, because it is incredibly important going forward, particularly in Year 13. When we get to Year 13, I'm going to expect that you already know a lot about ATP. So with that in mind, I'm going to reteach this. If you feel like you've already got ATP, perhaps you spent the time out of school revising it, making sure you've understood it, do skip through these slides and have a look at them and make sure you've got it all. But if you've understood the whole of ATP, you feel like you can answer the questions I'm going to pose, then feel free just to skip this topic and do some work on the work you needed to do earlier in the week or on other subjects. So today we're going to talk about the chemical structure of ATP, the bonds that are inside of it. We're going to describe how it's produced and also how we can hydrolyze it, and we're going to describe the enzymes involved. Now, for a start, I'm going to expect you to know a small amount of GCSE respiration here, but this is the only part of respiration I'm going to expect you to know. Excuse me. There we are. Glucose and oxygen, carbon dioxide and water. If you know that word equation, that's all I need you to know about respiration at this point. In Year 13, we will go into an enormous amount more detail about all of the different parts of respiration. But for now, simply learn this word equation, which you should already know. If you don't know it, you need to learn it. And as part of that, we're also going to release a small amount of energy. And the question we should have is, what is this energy? Excuse me, my computer seems to be having a bad day today. Now, this energy comes in the form of a chemical called ATP. And when we break down the glucose into smaller molecules, and specifically carbon dioxide and water, that releases a small amount of chemical energy and creates this ATP. I know there will be a few chemists out there who are screaming at their computers, but sir, when you break a bond, you don't release energy, you require energy, it intakes energy. And the answer is to this question that you actually are making more bonds than you are breaking. And so you are releasing energy overall. But regardless, forget all the chemistry for now. When you break down glucose and you use the oxygen to make carbon dioxide and water, it will release energy which will create this thing called ATP. Now ATP is very linked to a generalized nucleotide. So what I'd like you to do in your books, and I will give you 1 minute for this, is I would like you to draw me a generalized nucleotide from the shapes that I've just given you. I'd like you to label it and tell me what each bit would be. What would the circle be? What would the pentagon be? And what would the rectangle be? This should be very familiar from the nucleic acids topic. So I have 1 minute starting now. So that was just under a minute, but hopefully you've had the time to do it. Now, what you should have drawn is something that has a circle attached directly to the pentagon. The circle should be a base. Excuse me, my computer seems to be having all kinds of issues this evening. The circle, should not be a base. The circle should be a phosphate. The sugar in the middle would be the pentagon, and the rectangle would be the base. Now in this case, we're going to call that base adenine. So this is an A nucleotide. Now what I want you to think about is, if I wanted to create something that looked like adenosine triphosphate, what would the differences be? So you've just drawn something which is an adenine nucleotide. What would adenosine triphosphate look like? What would the difference be there? Hopefully in your mind you're thinking, well, it would look exactly the same, but it would have 3 phosphates, and you would be exactly right. What you effectively create when you put an A base on a pentagon of ribose is you create something called adenosine monophosphate. That is the proper name for the nucleotide containing adenine. If you want to create adenosine triphosphate, it's exactly the same molecule, it just contains 3 phosphates. So effectively, ATP is an adapted nucleotide. So that is ATP. Now have a minute again. You can draw out a simplified version of this just using circles, pentagons, and bases, if that will be helpful to you. And then can you label 1, 2, and 3? So that was a minute and a half-ish. If you've not had time to draw the molecule, just pause me. What you should be finding is that part 1 is 3 phosphate groups. It's the phosphate groups. So you can see that because it's got a phosphorus in the middle, and then the oxygens and the hydroxyl groups coming off. Now you're not expected to be able to draw a phosphate group, you are expected to recognize one when you see it. So there's 3 phosphate groups all joined together there. Part 2, you can call it a pentose sugar. In actual fact, that's ribose. You can see it's ribose because it's got the oxygen at the top, so it's definitely not deoxyribose, but I wouldn't expect you to recognize that. But it is ribose, so ATP is containing ribose. And then part 3 is a base. It's a nitrogenous base. Specifically, it's the A base. It's the adenine base. So ATP is made of 3 phosphates, a ribose sugar, and an A adenine base. And that's why it's called adenosine triphosphate. There we go. Now, we call it a nucleotide derivative, and you can take down these notes if it will be helpful to you. It's a nucleotide derivative because it's created from a nucleotide. And that's pretty much what I've just said, to be fair. It's used to store energy in cells. Now, I say store energy in cells, it's perhaps slightly misworded. It's used to release energy in cells. We don't store our energy as ATP, we store our energy in the form of starch in plants and glycogen in humans. We store that glucose for the long term, and that can then be used to release ATP, and that ATP can be used to release energy. So perhaps better to word that as it is used to release energy in cells. There is no other form of energy release which we need to know about. ATP is the sole form of energy release. So when you're doing respiration, and in GCSE we learned we released energy, in actual fact what we're releasing is this chemical called ATP. Now, you're expected to be able to think of a few uses of energy in the cell. Now those are quite straightforward, and I'm sure in your head you've already got some in mind. Something along the lines of movement, active transport. That's the kind of things we should be thinking about. Now, the next question is, from your GCSE, how is energy stored in this chemical? So I'm saying ATP is got energy in it. Where is the energy in this chemical? And how do you release it? Hopefully, you immediately think of bonds. The bonds are where the energy is stored. And so in order to get this energy out, we need to change, in actual fact, we need to make more bonds. Now, ATP is an incredibly unstable molecule. Naturally, it comes in the form of AMP, adenosine monophosphate, which is the nucleotide we were talking about. And what that means is it's very easy to break the bonds and to create adenosine diphosphate with 2 phosphates and adenosine monophosphate with 1 phosphate. Now, when we add water, we do a reaction. So what I'd like you to be thinking about, I'd like you to write down perhaps, is firstly, what would be the equation for this reaction? So what do you think is going to be created when I add ATP and 1 water molecule together? And secondly, I'm going to tell you it's going to break this molecule down slightly. What kind of reaction is that? What kind of reaction is it when we break a molecule using water? And we should have that right on the tip of our tongues. So I have 30 seconds. What's the equation for this reaction? And what type of reaction is it? So, hopefully, you should have said that we're going to turn ATP and water into ADP and what we're going to call PI, an inorganic phosphate, a phosphate. So you say ATP and water turns to ADP and a phosphate group. We're not breaking it down into AMP. If you wrote we're going to break it down to AMP because we only have 1 water molecule, so we can only break 1 bond. I'm going to break this bond here. We're going to create ADP and an inorganic phosphate. Now, the type of reaction is a hydrolysis reaction. And so we need an enzyme for this to happen, and that enzyme is called ATP hydrolase. The clue's in the name, ATP hydrolase hydrolyzes the bond. And that releases ADP and inorganic phosphate. So get down the name of that enzyme. You need to know ATP hydrolase is the enzyme that breaks ATP down. And the reason I break the ATP down is because when I break it down, it releases energy. For those chemists out there, it releases energy because this will then form bonds with the water around it. The ADP will form more bonds with the water around it. Those bonds actually have a higher bond enthalpy than the bond with the phosphate, and so it releases energy overall. For those of you who don't like chemistry, don't worry about it. Now, that will release something along the lines of 30.5 kilojoules of energy, which is actually a fair amount for such a small molecule. Now, there's your key equation. ATP and water goes to ADP and inorganic phosphate. You're going to see PI in a lot of places. PI is the universal inorganic phosphate shortening. Now, obviously, that is not a chemical symbol. It does contain phosphorus, but it contains oxygens and hydrogens as well. We just use PI as a shortening. So that's not a balanced chemical equation, but it's the best we're going to do for now. 20 seconds to write that down. So, the question then is, how is ATP synthesized? Now, we're going to synthesize the ATP during respiration. And during respiration, ATP is created using an enzyme called ATP synthase. Now we need a huge amount of energy, in fact we need 30.5 kilojoules of energy to create an ATP. It's going to be a condensation reaction, so we're going to release some water as well, and we're going to go ATP and inorganic phosphate goes to ATP and water, which is just the exact opposite of what we just talked about. And now we need an enzyme called ATP synthase because it is synthesizing ATP. And when we come back in year 13 to talk about respiration, ATP synthase is going to be the key enzyme we're going to talk about. So we're going to talk about ATP synthase in a lot of detail. But for now, all you need to know about it is it's turning ADP and inorganic phosphate into ATP and water. And because we're adding a phosphate group, we're going to call that a phosphorylation reaction. Again, we're going to learn about a lot of phosphorylation reactions. So this is the first one we're talking about. But this is a phosphorylation reaction. Again, I'll give you 40 seconds to copy this down. If you find that I'm moving too fast, do feel free to pause me, I wouldn't mind. Right then, let's move on slightly. Now, there are 3 types of phosphorylation reactions which we're going to use to make ATP. We don't need to worry about any of them right now. I just think it's worth bearing in mind what they are, because when we come in year 13 to talk about photosynthesis and respiration, we're going to be talking about these in a lot of detail. So you may come across these terms when you're going through your textbooks. I don't even know that it's worth you writing them down at this point, because we will talk about them a lot in year 13, and they won't come up in your paper 1. But very broadly, photophosphorylation happens during photosynthesis. So when you are doing photosynthesis, you have to release a little bit of ATP in the middle of that reaction to make it keep going. Oxidative phosphorylation happens during respiration. Again, that's using ATP synthase to release ATP for energy usage. And then substrate level phosphorylation is where we deliberately transfer phosphate groups directly onto ADP, from other molecules. And again, that will be happening in respiration a small amount. But for now, I would say don't worry about this. Only bring it up in case somebody mentions these terms to you in passing. For instance, a teacher, I think oxidative phosphorylation was talked about in your lesson, or if you come across it in a textbook. Now, the human body contains a very small amount of ATP. And as it says in the PowerPoint, about 5 to 10 seconds worth. However, we need an enormous amount of ATP. Every day, you use up your own body weight in ATP. So how is that working? What are you thinking? How could that possibly be? Well, the answer is that you are constantly making it. In fact, it is an immediate energy source. You can't store it for long periods of time. And why not? Well, hopefully you're thinking back to the slide we did earlier where we talked about how it's a very unstable molecule. That bond is very easily broken. It doesn't require much energy at all. And so it fairly much spontaneously breaks down of its own accord pretty quickly. This is also why you die very suddenly if you take something like cyanide, which is a respiration blocker. You don't have any ATP stored up. So if you take something that stops respiration and you stop making ATP, you run out pretty much immediately. And when you run out of ATP, you run out of energy. When you run out of energy, your heart stops beating. And that's how these poisons work. Now, glucose has an enormous amount of energy in it. But ATP is a better immediate source of energy than glucose because it releases less energy, but it releases it in smaller, more manageable quantities. You don't need much energy to run an active transport protein. If you had a molecule of glucose running around, releasing all of its energy at once, it would be far too much to power an individual active transport molecule, and so you would waste a lot of energy. It's also much faster than releasing the energy from glucose. It's a single step, so that I can release this small amount of energy very quickly, directly where it's needed. Now you do waste some energy in the glucose. The glucose is not very efficiently broken down into ATP. Excuse me, let me re-say that. The energy from glucose is not very efficiently transferred into the ATP. But it is worth it so as to have these individual small pieces that can be easily broken down quickly without having to wait for the glucose to be digested. I'm aware that my camera box has gone over the top of that note, so let me put it up there. Right, hopefully that's enough time to take the notes. Pause me if not. Now, the why do organisms need energy thing is technically on the A level. I think it's quite patronising. Hopefully, you can think of a lot of reasons. Now, we talked about a couple earlier. Now, there are the examples. Metabolic processes, movement, active transport, repair, division, maintenance of cells, secretions, maintenance of body temperatures, activation of molecules by phosphorylation. Now, can you write down those for me? You are expected to know them. Some of them are perhaps not exactly obvious, but I think most of them are quite straightforward. So what I need you to do then is to think, for the metabolic processes, what am I talking about? For active transport, what kind of thing could I be actively transporting? For secretion, what kind of thing am I secreting? So have a couple of minutes just to write that down. What do I mean by metabolic processes? What do I mean by active transport? What kind of examples can I think of? Have a couple of minutes for that. I'll just turn my camera off and allow you to get on. Hopefully that's enough time for you. So when we're thinking about metabolic processes, I would like you to be writing down some thoughts as you go along, so I'm not just to give you all the answers, but we're thinking about things that are involving the usage of organic molecules. Things like building glucose into larger carbohydrates. Things like the digestion of other molecules. These things will require ATP. Movement, fairly obvious. Active transport, well, you could be thinking about actively transporting, for instance, ions out of the soil by root hair cells. So we're not just talking about humans here. Every animal uses ATP, and every plant uses ATP, and everything that is alive uses ATP as far as we're aware. So, active transport, ions out of the soil into root hair cells. Secretion, You could be secreting a lot of things. Hormones, you could be secreting enzymes for external use. That requires energy in order to transport them out of the cell. Maintenance of body temperature only occurs in endotherms, obviously things that create their own body heat. Lizards that are basking in the sun don't require the ATP to do that. And then the activation of molecules by phosphorylation. We're going to come back to that in a lot of detail in year 13, but sometimes when you want to turn on, for instance, some DNA, you do that by attaching a phosphate group. That phosphate group can come directly from ATP, and so the ATP can be used as a donor of phosphate, if you like, for that to happen. Now, come back to that one later. I would focus on movement, active transport, division, repair, because those are the really obvious ones that hopefully everyone can remember. Right, that is the end of that. Now I'm going to skip these questions. If you want to pause and have a look at the progress check, you have a couple of minutes just to do that. Do it. I would do it in class, but I don't think there's much point me sitting and watching you do that. What I'll do is I'll go on to the exam question, which I do want to do with you, and then I'll come back and give you the answer to these later. So if you want to do these questions, just pause the video now, do the questions, and I'll give you the answers after the exam question we're about to do. Okay, now this is something, again, I'll move myself. This is, oops, hopefully nobody paused over the answers there. This is something that comes up rarely in exams, but it can. Now this is a 2012 example, which told you how often this comes up in exams. ATP is useful in many biological processes. Explain why. 4 marks. Now that's 4 minutes. I would like you just to sit down, have a think about what are the possible bullet points. If you did see the answers, I'll apologise. One of those things. Okay, so 4 minutes starting now. Right, so that was 4 minutes. Now, that's quite useful to see because that's the amount of time you'll have to answer these questions in an exam. In actual fact, you have just over 4 minutes in an exam. But if you've got it done in less time than that, if you've had to skip me forward, then be aware that you have the time just to go back and do some more work on these questions, or actually to go back and do the more difficult questions that come around graphs and data. What I don't want is for anybody to be stressed at any point about timings for exams, because there's no need. You will have plenty of time. And if you need extra time, you definitely can get it, but I don't think you will. Mostly because this is nice, quick, easy questions, this kind of question. Now, as far as the mark scheme goes, it focuses mainly on how ATP is used. So, release energy in a small, manageable amounts. That's a really, really important fact about ATP. In fact, it's why it is useful. It's broken down in a single step, or a single bond broken, or it makes energy easily available. Well, that's also true and it's very useful. The fact that it can add phosphate to, for instance, nucleic acids, that is also true and potentially useful. I wouldn't necessarily include that point in year 12, but if you can remember it, you definitely should. That it lowers the activation energy for other activities, and that it can be easily reformed and made again. Now, points 3 and 4 are points we're going to cover in a lot of detail in year 13. So as long as you're getting points 1, 2, and 5, then at this point I'm very happy. Because by the end of year 13, points 3 and 4 will be very obvious to you, and it'll be one of those things that it's going to duh moments. Yeah, I already knew that. But for now, 1, 2, and 5. 5 is the one I think people forget. Reformed and made again. That is important. If you have to constantly take in new food, for instance, let's say I had to eat new food in order to supply me with new ADPs and inorganic phosphates, then it would take a lot more energy in order to produce these things. The fact is that when you break down the ATP, you can immediately reform it again, as we talked about earlier. So we said we can break it down with ATP hydrolase, and you can reform it with ATP synthase if you have a bit of energy to put in. So you don't waste the products. The products can be reused again. Now, if I'm not wrong, yeah, that's the final slide. Now let me give you the answers to these questions if you did them. ATP stands for adenosine triphosphate. The different parts that make up ATP are 3 phosphate groups, an adenine base, and a ribose sugar. When ATP is broken down, it produces ADP and inorganic phosphate, and that is a hydrolysis reaction. And the kind of enzyme that causes the breakdown of ATP is ATP hydrolase. Now, you should know from today then what the structure and chemical makeup of ATP is, what it's made out of, how it's produced and hydrolyzed, and what enzymes are used. And if you've got that, then you've got ATP, and I hope that's been very useful for you. If there is any difficulty there, if you have any questions, contact me on Teams. I have no issue with helping people. I have noticed some questions about measles and STAR being brought up. I'm actually going to create a small resource about that. So if you've got questions about measles and STAR, we will go over it. Please don't worry about it at this point. All right. Looking forward to seeing you all in school, hopefully, the week after next. But week after next for me, probably next week for you. But whenever that is, it'll be good to see you all. And I hope you have a great week.